Hello, students. Have you ever wondered how detectives solve crimes with just a small sample of DNA? Or how scientists can tell who is your biological parent without a doubt? Welcome to the fascinating world of DNA fingerprinting, a technique so powerful that it can identify individuals with just a tiny trace of DNA. It's all based on the unique genetic code that makes each of us distinct, even though we are 99% similar to each other. Pretty amazing, right? As we dive deeper, think about this. Have you ever wondered how we all have different physical features, like eye color or height? Well, it's because of the specific sequences of DNA in our bodies that make us unique. DNA fingerprinting zooms in on these sequences and uses them to tell you exactly who you are, just like a biological barcode. Now, let's explore the key players in DNA fingerprinting. These are the repetitive DNA sequences. Picture this: if you look closely at your DNA, you'll find regions that repeat over and over again. These regions don't code for any proteins, but they vary wildly from one individual to another. Some of these repeats are so unique, they help scientists tell one person's DNA from another's with pinpoint accuracy. These repetitive regions, like microsatellites and mini satellites, are the real stars of DNA fingerprinting. Think of them as the genetic equivalent of fingerprints. The more repeats a region has, the more distinct it becomes. So, scientists target these sequences to make a unique genetic map for each individual. As we dive deeper, think about this. Have you ever wondered how we all have different physical features, like eye color or height? Well, it's because of the specific sequences of DNA in our bodies that make us unique. DNA fingerprinting zooms in on these sequences and uses them to tell you exactly who you are, just like a biological barcode.
Now, let's break down the process of DNA fingerprinting step by step. The first step is extraction. Scientists start by taking a sample, blood, hair, or even saliva, and isolating the DNA. Imagine you're pulling out a single thread from a giant ball of yarn. Then comes digestion. This is where scientists use special scissors called restriction endonucleases to cut the DNA into smaller fragments. It's like cutting a long piece of string into shorter, manageable pieces. Next, these fragments are separated using electrophoresis. Picture this: a giant gel slab where the DNA pieces race through like athletes on a track. The smaller pieces move faster, and the larger pieces move slower, creating a beautiful separation of DNA fragments based on size. After the separation, the fragments are transferred to a membrane, and we move on to blotting and hybridization. Think of this as transferring the raised results onto a scoreboard and marking the DNA sequences using a special probe that lights up when it binds to the target DNA dot. Finally, the autoradiography step visualizes the DNA pattern, revealing a unique banding pattern that belongs only to that individual. One of the coolest parts of DNA fingerprinting involves VNTRs, or variable number tandem repeats. These are regions where a short DNA sequence repeats many times, and the number of repeats varies greatly between individuals. This variability is what makes VNTRs so useful in DNA fingerprinting. Imagine a string of beads, each bead representing a repeating DNA sequence. Some people might have a long string with many beads, while others may have a shorter string. The unique bead pattern, based on the number of repeats, is what makes you stand out in a crowd of billions. Scientists focus on these VNTRs because they exhibit high polymorphism, meaning they're highly variable across individuals. So, when we compare the VNTR patterns from two people, they often look completely different, just like matching fingerprints.
Let's explore how PCR enhances DNA fingerprinting, a critical tool in modern science. First, PCR introduction, polymerase chain reaction. or PCR, has truly revolutionized DNA fingerprinting. It allows scientists to take even the smallest sample of DNA, sometimes just a single cell, and amplify it to make enough copies for detailed analysis. This has opened doors to analyzing DNA when only trace amounts are available. How PCR works? PCR operates through a simple yet powerful cycle of heating and cooling the DNA sample. This process causes the DNA to denature, then anneal with primers, and finally extend, making millions of copies of the specific DNA region under investigation. PCR benefits. The beauty of PCR lies in its efficiency. It allows DNA fingerprinting to be done faster and with much smaller or degraded samples, which is why it's become an indispensable tool in forensic science. It has dramatically sped up the process, enabling accurate results even from limited evidence. Real-life applications of PCR in DNA fingerprinting. In the real world, PCR is used extensively in criminal investigations, paternity testing, and identifying genetic disorders. It has transformed forensic science, enabling authorities to match DNA from crime scenes to suspects, even when the DNA sample is degraded or minimal. PCR truly makes it possible to connect the dots in ways that were previously unimaginable.
Now, let's talk about the many ways DNA fingerprinting impacts our world. In forensic science, it's a vital tool for solving crimes. Imagine finding a tiny drop of blood at a crime scene. With DNA fingerprinting, you can determine whose blood it is and solve the mystery in a matter of hours. But that's not all. DNA fingerprinting is also used in paternity testing. It can confirm a biological relationship between a parent and a child by comparing their DNA patterns. It's the ultimate proof of a genetic connection. Beyond crime solving, DNA fingerprinting helps scientists study genetic diversity within populations and monitor endangered species. It's also used in genetic counseling, helping to identify genetic disorders that might run in families. As we wrap up, let's revisit what we've learned today. First, we explored how DNA fingerprinting works, from identifying polymorphisms in repetitive DNA to the steps involved in the process. We also saw how the revolutionary polymerase chain reaction, PCR, has significantly advanced the technique, making it more accurate and accessible. Next, we looked at the powerful applications of DNA fingerprinting. Whether it's solving crimes at a scene, identifying family connections, or even tracing ancestry, this technique has become an indispensable tool with implications that extend far beyond science. In the realm of scientific impact, DNA fingerprinting has played a crucial role in genetic breakthroughs. It's enabled researchers to map human genomes and pinpoint genetic disorders, revolutionizing the field of medical genetics and providing new insights into human biology. However, with all its power, we must also consider the ethical implications. DNA fingerprinting raises important concerns about privacy, consent, and its potential misuse, especially in legal contexts. These issues underscore the need for responsible use and strict guidelines in implementing this technology. And there you have it, the amazing science behind DNA fingerprinting and the lasting impact it has on our world. Thanks for watching, and we hope this video has given you a deeper understanding of this incredible forensic tool.